My name is Katherine Do, and welcome to the last video for the Clinical Metaproteomics tutorial. In this video, we will cover Module 5, Data Interpretation. Previous videos covered background in Modules 1 through 4, so please watch those to prepare for this video. Now, let's begin. First, we will summarize the quantification module and how it set us up for this module. Then, in the hands-on section, we will execute the data interpretation workflow, where we will perform taxonomic and functional annotation using Unipept, followed by statistical analysis using MSstatsTMT. Here is the recap of the quantification module. We used MaxQuant to quantify verified peptides and selected for distinct microbial peptides from the peptides file. We also generated evidence and protein group files. These files will come into play for this module. Next, let's see where we are in the Clinical Metaproteomics tutorial. The previous videos have covered database generation, discovery, verification, and quantification. The focus of this video is data interpretation. For taxonomic and functional annotation, we will use Unipept as it is capable of analyzing mass spectrometry-based proteomics data to study microbial communities. Then for statistical analysis, we will use MSstatsTMT. If you remember from our overview video, I mentioned that during sample processing, proteins were isolated, digested with trypsin, and labeled with a distinct tandem mass tag, or TMT, tagging reagent. TMT labeling enables simultaneous identification and quantification of proteins across multiple samples. The MSstatsTMT tool was designed for robust statistical analysis of mass spectrometry-based quantitative proteomics data using TMT labeling. Next is the hands-on section where we will execute the data interpretation module. Here I have already prepared the history with the inputs and imported the workflow. We can select the inputs and then click Run Workflow. The list of quantified peptides has been filtered for microbial peptides. The evidence and protein groups files have not been filtered like that, so we will be able to visualize and compare human and microbial proteins using them. Here is the overview of the data interpretation module. The list of quantified microbial peptides will be put into Unipept for taxonomic and functional annotation. The reason we use Unipept for this step is because it is a powerful bioinformatics platform that integrates with public databases like Uniprot to perform taxonomic and functional analysis. This facilitates the identification and quantification of microorganisms within diverse environments using comprehensive and up-to-date information for annotations. Unipept also allows for comparative studies across samples, conditions, or time points, which can give insights into the dynamic responses of microbial communities in response to environmental changes. Following this, the MaxQuant evidence and protein groups files will be put into MSstats for statistical analysis. This tool allows users to define the experimental design, including sample groups and conditions. MSstatsTMT outputs will provide useful information about differential protein expression across conditions, estimates of fold changes, and associated p-values. These findings have the potential to aid in the identification of biologically significant proteins. We will be able to visualize and compare human and microbial results. Now that we have some results that go beyond the number of peptides and proteins, I want to reiterate again that this tutorial material is meant to highlight the effectiveness of our clinical metaproteomics workflow. We used a small example set of quantitative proteomics data from an ongoing study of ovarian cancer in fluid from cervical swabs. Because the sample data is so small, Results in this tutorial are not meant to bear any sort of biological significance or communicate any new upcoming breakthroughs. When I mention that a certain genus is the most abundant, or that a protein acts as an enzyme to catalyze a certain chemical reaction, I am specifically discussing them in the context of this demonstration. Confirming the presence of identified microbes and their pathogenic potential will require further validation that is beyond the scope of this tutorial. However, we hope that by the end of this tutorial, we have successfully demonstrated the potential of this bioinformatics pipeline to enable new discoveries in clinical research. With that out of the way, let's talk about tools. First up is Unipept. Unipept will perform taxonomic and functional annotations, and the two microbial outputs that we will discuss here are the taxonomy tree and enzyme commission proteins tree. You are welcome to explore the other outputs. Here I will show you how to view the microbial taxonomy tree, and on the next slide I will discuss how Unipept generated it, as well as how to interpret it. I will now switch to a prepared galaxy history to show you how to view your outputs. Not only does Unipept give visual outputs, you can also view the data in tables. To do this, find the file called Unipept-pept-info and click on the eye icon in the top right corner. You can see each peptide on its own row and scroll through the columns for more information. Find the microbial taxonomy tree and click to open the options. We will click on the icon that shows a small graph. If you hover your mouse over it, the word Visualize will pop up. Other outputs will have different options, but here we want to click on Unipept Taxonomy Viewer. This will give you 3 interactive views to choose from: Tree View, Sunburst, and Tree Map. Choose the one that makes the most sense to you. Personally, I tend to use the tree view because the node or the circle size reflects how many sequences were identified at or lower than that level. You can zoom in and out, drag to pan, etc. When I click on a node, I can see the taxonomic branches sprout, and when I hover over a node, I can see how many sequences were identified at that level. So, how did Unipept generate this tree? Unipept determined taxonomic composition by performing LCA, or lowest common ancestor analysis, on the microbial peptide sequences. The Unipept tool takes tryptic peptides as inputs, and for each given peptide, Unipept retrieves UniProt entries. These entries include accession numbers, as well as a complete set of organisms in which the peptide occurs. The set of organisms for each peptide is processed using the Unipept LCA algorithm to obtain the most specific taxonomic rank that the organisms share. In this workflow, Unipept LCA analysis allowed for the assignment of the most likely genus and species whenever possible, as well as functional categories for the 155 quantified microbial peptides. On this slide, you can see that the taxonomy tree depicts the likely taxonomic assignments for each peptide sequence. From our example datasets, we found that Lactobacillus was the most abundant genus, followed by Escherichia and Citrobacter. Next, I will cover the microbial EC, or enzyme commission proteins tree. You can view it the same way you did for the taxonomy tree. First, find the output, click on the visual option, and click Unipept Taxonomy Viewer. So, what is an enzyme commission proteins tree? This tree displays the numerical classifications known as enzyme commission numbers, or EC numbers, of what chemical reactions are catalyzed by proteins that function as enzymes. These numerical codes do not name specific individual enzymes, but rather identify classes of enzymes that catalyze similar reactions. Again, like the taxonomy tree, you can zoom in and out, click on nodes to open more branches, hover to see how many sequences are specific to a level. This proteins tree visualizes the 7 classes of chemical reactions. The node sizes also reflect how many sequences belong to a certain class. On this slide, you can see that the most prominent classification was Class 2 for transferases, followed by Class 3 for hydrolases and 4 for lyases. To know the chemical reaction by peptide, you can view the output called microbial EC terms output. Be aware that some peptides may share the same EC number and name, as they may catalyze similar chemical reactions. You may also notice that not all of the 155 microbial peptides will have an EC number. This could be due to the fact that not all chemical reactions have been assigned a corresponding EC number, or some proteins in this data just do not act as enzymes at all. Next, we will move on to statistical analysis. For this, we will use the MSstatsTMT tool. Here, we will analyze human and microbial protein groups separately. To do this, we can simply filter the proteins by their tags. Human proteins have a tag that says _human, while contaminant proteins have a tag that says _. The remaining proteins with microbial taxonomy tags are, of course, designated as microbial proteins. At the time of this video, we obtained 1,117 human proteins and 61 microbial proteins. Again, if we were working in the real world with plenty of sample data, these numbers would be different, but even with this limited amount of data, we can observe the high abundance of human proteins compared to how few microbial proteins were present. So, what is MSstatsTMT? Remember that the example datasets were labeled with TMT reagents for quantitative proteomics. If you are not working with TMT, the MSstats package itself is compatible with label-free quantitative proteomics data. The MSstatsTMT tool is compatible with data processing tools such as MaxQuant and allows for sensitive and specific detection of differentially expressed proteins in large-scale experiments with multiple biological samples. This tool does require an annotation file and a comparison matrix file. The comparison matrix can sometimes be referred to as a contrast matrix as well. We have provided these files for convenience. The annotation file dictates how the quantifications will be combined, and the comparison matrix or contrast matrix will accommodate the 3 different sample groups, which are ovarian cancer, healthy, and benign. Using these inputs, MSstatsTMT removes all of the proteins labeled as contaminants from the MaxQuant protein groups file and performs statistical analysis to discern the differential expression of quantified proteins across the 3 sample groups. This allows us to generate tabular files that contain protein abundance ratios, group comparisons, and volcano plots, which are able to showcase differentially expressed proteins between human and microbial protein groups. In this video, we will focus on the volcano plots and comparison plots, but you can explore the other outputs on your own. The file named Group Comparison will contain the statistical results that we will model. Similar to when we discussed the Unipept taxonomy tree and EC proteins tree, we will focus on the microbial, volcano, and comparison plots. I will briefly show the human plots just to have a visual representation of how much more abundant the human proteins are compared to microbial proteins. The volcano and comparison plots will be in PDF format. So, to view them, instead of going through the Visualize button, you will press the eye icon. Each PDF contains multiple plots, so I have selected a few plots as examples to talk through how to interpret them. Again, we are not claiming any biological significance or new breakthroughs. Here are the 4 plots I will discuss. The top 2 plots are for microbial proteins, and the bottom 2 are for human proteins. We have the volcano plots on the left and comparison plots on the right. I'm going to temporarily cover the comparison plots while I discuss the volcano plots. A volcano plot visualizes the log-fold changes and negative log 10 of adjusted p-values for a comparison between two sample groups, also known as case groups, for the quantified proteins. Here we will compare ovarian cancer versus benign cases for the 61 microbial proteins. To put it more simply, for each protein, we will compare its abundance in one sample group, like in ovarian cancer cases, to its abundance in another sample group, like in benign cases. Ultimately, we want to determine if the difference between these two groups, if any, is statistically significant. For the volcano plots, each data point represents a single protein. As you can see, there are many more proteins for the human plot. Remember that we obtained 61 microbial proteins compared to 1,117 human proteins. I do want to note that the human volcano plot I included here doesn't have the protein names like the microbial plot does. I prepared the human plot to hide the protein names as there are so many that they would overlap and just be unreadable. The workflow we provided should keep the human protein names on these plots when you run it on your own. Additionally, the data point color denotes any differential abundance between cases. The legend is also shown at the bottom of the plot. Gray represents no regulation. Then we have blue. This represents down regulation. We also have red, which represents up regulation. In our example plot here, comparing ovarian cancer and benign cases, we can see 3 statistically significant data points. They are blue, which means that these proteins are downregulated in ovarian cancer cases compared to benign cases. This plot does not have any red or upregulated proteins, but we can quickly see some examples in the human volcano plot. These red data points represent human proteins that are upregulated in ovarian cancer cases compared to benign ones. Circling back to microbial proteins, the last plot we will discuss is the comparison plot for this microbial protein here. For the comparison plots, instead of comparing multiple proteins as we do with volcano plots, a comparison plot visualizes log 2 fold changes and variation of multiple comparisons for a single protein. This plot allows us to see how a protein can be differentially expressed across different sample groups. The data point for the ovarian cancer to benign comparison is below the horizontal dashed line, which equals a decrease in protein expression. This corroborates what we saw previously. This protein is downregulated in ovarian cancer cases compared to benign. This is the end of Module 5. To wrap up this tutorial, let's review the modules one last time to see how much we have accomplished. Starting from the beginning in Module 1 Database Generation, we generated a customized database for clinical metaproteomics that comprised of protein sequences from our many species of interest, humans, and contaminants. Then, in Module 2 Discovery, we searched MS datasets against the reduced database from Module 1 to identify peptides and proteins. Then, in Module 3 Verification, we used PepQuery 2 to verify the presence of peptides as well as validate that they are indeed of microbial origin. In Module 4 Quantification, we performed quantification of microbial peptides. Lastly, in this video for Module 5 Data Interpretation, we looked at the taxonomic and functional annotations of the quantified peptides using Unipept, and we performed statistical analysis using MSstatsTMT to identify proteins that were differentially expressed across different sample groups. This marks the end of the Clinical Metaproteomics tutorial. Congratulations on completing this tutorial. We hope that it was enlightening for you and proves to be useful, whether you are a beginner or an experienced researcher. We welcome feedback on this tutorial and these videos. Please let us know what you liked and any suggestions for how we could improve. Lastly, thank you so much from the Galaxy Proteomics team, and we hope you continue to explore other tutorials on the Galaxy Training Network.